Now I would like to invite Dr. Noshin Ilyas, Assistant Professor, Arid Agricultural University.
I'm Dr. Noshin Ilyas, working as an assistant professor in the Department of Botany, primarily Arid Agriculture University, Rawalpindi. And today I'm going to discuss some of the work which is being carried out in my lab upon the plant-microbe interaction for alleviating the abiotic stress. I hope we all know from that we belong to an agricultural country, and our main sustenance, the whole economy of our country, it is depending upon the agriculture. And these are the things which are very well known to us that for example, when we say that our 18.9% GDP that is coming from the agriculture, it is providing food to a large number of population. Along with it, it is the main earning source, and we have a large cultivation for the crops. There are many crops which we are producing. Main important are the wheat, rice, cotton, and sugarcane. But when we are talking about agriculture in Pakistan, there are some plus points and there are some threats. For example, this agriculture, this is playing a very important role in our industry, in foreign exchange, and also for our industrial good and machinery. The important things are, the interesting opportunities are that we have a great diversity. We are a very diverse country, not only ethnically and linguistically, but also depending upon the area. From one side, we have the sea levels, and the other side, we have the highest mountains of the world. It also means that we have diverse environments. We also have the harsh environmental or extreme environmental conditions in our country. We are also one of the most populated countries. We are the sixth most populous country of the world. So we have a large population to feed, and this population is also increasing with every passing day. And this all is creating some food issues, food security issues in our country. This picture is very important. This slide is very important because here we are discussing what is happening in Pakistan and what are the predictions for Pakistan. We are a country which are supposed to have the hunger severity index, means our population is underfed. We have to feed a large amount of the population. The other important crisis which we are facing these days, that is the water. We are not the countries which have the shortage of the water. Rather, we are among the countries which do not have any water. We are included among the waterless country. We only have 2.56% of the water which is available, and that is very less as compared to our demands. Along with it, we all know that we are a country that is vulnerable to the climatic change. We are the seventh country which has been considered to be vulnerable to the climatic change. And this means the unexpected weather conditions, the anomalies, and the mishaps and the certain weather condition change. And this is affecting our agriculture because the agriculture, the crop production is dependent upon the season. It is dependent upon the environmental climate. All of this, it is impacting on our agriculture, and this is a very serious kind of prediction. And it has been predicted that our agriculture production, due to all these conditions, may decrease in the future. Though we have an increasing population, but due to all these conditions, if we do not look into it, we might face a decline in our agriculture production. And this estimate is up to minus 15, so minus 25% has been forecasted for our country, which is a serious and alarming situation. Now, along with it, we have very less sustainable agriculture in production. Most of our agriculture production that is dependent upon the normal chemical fertilizers. And we also know that our soils, they are facing a nutrient deficiency kind of situation. We are most importantly deficient in the organic matter. We have 100% deficiency of nitrogen and organic matter. And we also have the deficiency of the major macronutrients in our soils. And we also have a very high fertilizer use efficiency, means as compared to our neighboring countries, in our country, the crops, they are using more fertilizer per production rate, and this is causing more and more fertilizer consumption. Now, what is being done in my lab, or what we are doing, we have divided this work into three parts. First of all, we are studying the plants for the stress tolerance mechanism. Until and unless we don't know what is happening in plant, how a plant is combating the stress, how it is alleviating its stress, mitigation mechanisms in the plant, we are not able to formulate any strategy for the plant stresses. Along with it, we are working upon the soil amendments and soil microbes. And in the third, we are combining these things and we are studying the plant responses. So this is the research work. This is the basic scheme of the research work, which is being carried out in our lab. In the first part, it is divided into three parts. In the first part, we study the plant for the stress tolerance mechanism. In the second part, we go for the isolation of the indigenous BGPR from the stressed ecosystem. Why? Because they may help in the selection of the stress adapted strains. Then we characterize this PGPR. We have done some biological, physiological, and molecular characterization. And then we also, because the soil is a complex system, so we also have worked for some amendments like compost, biochar, and some even have tried the alternate cropping system. In the third part, we are combining these things. We are combining the microbial strain, the amendments, and we are applying them on the plants, particularly under the stress conditions, and we are studying the plant responses. And we are also trying to elucidate the molecular mechanism which might be involved in these stress tolerances. So this is what we are doing. We are targeting the organic amendments, we are targeting the microbiomes, microbial inoculations, and we are selecting the plants that leave this positive soil gases. And all of this, we are more, you can say, we are targeting a whole system rather than the individual components. I think there has been a lot of discussion about the PGPR, so I'll skip it. You know how they can improve the soil fertility and how they can help the plant growth. Similarly, we have the organic amendments. These are also important in improving the soil carbon, particularly the organic carbon. They are improving the soil physical properties, water holding capacities, the nutrient retention techniques, and also they have playing some role in the erosion, control of the erosions and increase in the microbiological activities. These are the main organic amendments we have used. Definitely, as this whole webinar is concerned with the microorganisms, so we have done some work on PGPR along with the amendments which we use. They are compost, biochar, and some. Now we are even working on the bioorganic fertilizer. These are the stresses which have been covered in my lab. We have done some work on the drought stress, salinity stress, heavy metal stress, hydrocarbon, and we have some work done on the environmental pollutant. This is always a long way to go. This is some example what we are doing in lab. I can provide the details to whosoever who wants some detail. In the first part here, you can see these are the QTL mapping. This is the QTL analysis. This is the QTL analysis of the wheat. You can see the 7 chromosomes, all the 3 genomes, A, B, C, A, B, D, A. And here we have marked the traits which might be responsible for the drought tolerance in the plant. And these traits, they provide an indication that these are the main things which are helping the plant in combating the stresses. So first of all, we did the QTL analysis of the wheat plant. Then we went for the second part of our work in which we isolated some strains from the stress regions. Here we targeted the arid regions of the Pakistan. And then we screened these strains for their tolerance, particularly in the different water potentials. We also screened them for their PGPR characteristics like indole-acetic acid production, ACCD minus, the normal, the phosphorus-solubilization, all these parameters, they are very familiar to all of us. Then we did some phenotypic and genotypic characterization. We have done some 16 RNA analysis and obviously submission to the NCBI, and the submission accession numbers have been obtained. These are some of the promising strains which have been isolated by my lab, and the most important, you can see the details. We have isolated from some of them from the salt ranges, from some of them from the drought affected regions, and particularly the arid regions of the Pakistan. And these have been submitted and accession numbers have been obtained. The important characteristic over here we have mentioned is that we have also checked the phytohormone production, and these are basically isolated, or you can say these have been selected based upon their ability to produce the abscisic acid. That's a stress hormone. So this is also some work that is being carried out that these hormones, these have been tested, for, their presence has been checked in the PGPR, which have been isolated from the control and the stress site, and we do the comparisons, and we have seen that the microbes which are isolated from the stress region, they have the ability that they have more production of the abscisic acid along with the other hormones, and this helps the plants particularly for combating the abiotic stresses like drought and salt stress. So this is the third study where we combined these PGPR inoculation and we applied the stress, and we tried to find out if these PGR have any ability to help the plant in mitigating its stress, and we got some very good result. This is the effect of azospirillum inoculation on maize and the drought stress, and we observed that the plant responses, particularly all the responses, we target 3 main responses in plant: the morphology, the physiology, and the biochemical parameters, and the antioxidants particularly. And we observed that these PGPR had some mechanism with the help of which they can improve the growth parameters of the plants, particularly under the stress condition. Along with it, we applied this PGPR with some soil amendments also, and we got some very good result. And these are the details of some of publication in this, particularly these are the details of the publication which we carried particularly for the drought stress. This is the some of the papers which we did for the part 1, that is where we studied the plant responses to the stress condition. The second part where we isolated the strains which have the ability to tolerate the stress condition, particularly the drought stress. And in the third part, we have combined these things together, and we have tried to find out if we are able to define some mechanism with the help of which we can improve the overall growth of the plant. Same way as I have discussed about the salt stress, we did some work on the, I have first mentioned about the drought stress. Here we have some work on the salt stress, and you can also see that these are the QTLs for the salt stress, where we did genome mapping of the traits which are responsible for the salt stress in the plants. And these are also the chromosomal maps for the wheat. And along with it, in the second part, we also isolated some strains. I have mentioned some of these strains in an earlier slide. And here we tested this strain for the production of the compatible solute. We tested the bacteria for the production of proline, reducing sugar total soluble content. And we also checked that if these strains are, how these are working in alone and how these are working together. And we found that the consortia is performing more better, and it is to have the ability to produce this compatible solute under the stress condition. So they might be a, you can say, a better inoculum to help the plant in mitigating the stress. So in the part 3rd, we studied the plant responses to PGPR inoculation and soil amendment, particularly under the salt stress condition. And this is the paper where we obtained the results that these amendments and the PGPR, they're helping in the salinity tolerance in wheat. And we have also done some studies for the other crops also. So these are the publications for from my lab for the work of the salinity. The first one where we have done the QTL mappings, and then we have isolated some rhizobacteria strains for the salinity tolerance. And in the third, we have applied them, and we studied the salinity tolerance in the wheat plant. The abiotic stress doesn't limit to the drought and the salinity. This is a whole ever expanding field. So we have tested some other effects also. For example, we have gone for the heavy metal, which is a rising pollutant in our environment. And we have done the same, we are following the same scheme. We have done some investigation on the effect of these heavy metals on the plants. And then we have, these are the whole genome sequencing of some of our bacterial strains, which have shown very good potential for the tolerance of the heavy metals. And after these strains, they were also tested for their inoculation abilities to help the plant in mitigating the heavy metal stress. And these are the publication work which we have done for the heavy metal tolerance. Some of the strains, they have shown good potential against like the chromium and effect. And along with it, we also tested their potential in combination with the biochar. And we have studied the amelioration potentials, and we have got some good results. Along with it, the main work, we have another kind of main work which we have carried in our lab, that is upon the isolation of the crude oil degrading plant growth promoting rhizobacteria. And in this work, we have been able to develop a plant microbiome remediation system for the crude oil contamination. And this was particularly done for the maize crop, and we got some good results from this one also. This has also been published by Journal of Environmental Chemical Engineering. This is a good journal. And this work was highly appreciated. We have also done some work on the plant growth promotion and the phytoremediation potential of pyrochar and the other amendments for particularly under the hydrocarbon pollution. We have done some work on the nanobiotechnology also, and we got some very good results, particularly for the silicon nano particles in combination with the plant growth promoting bacteria, and they help the plant in the drought tolerance of the plant. This work has been published in the Plant Physiology and Biochemistry. We have also done some the molecular analysis. We have done the quantitative proteomic analysis for copper nanoparticles also. So this is the latest work which we have carried. We have now worked for a production of a bioorganic fertilizer. This fertilizer has been prepared through the fermentation of the agro waste by the PGPR. And this work has led, this is basically where we have produced a product that is our bioorganic fertilizer, and we are now testing it under the field condition, and we are trying to upscale this, and this work has been also been published in Chemosphere. But we have found that we have to go for applications of the PGPR other than plant growth promotion. We have done some fermentations by using them, which are doing two-way good. They are not only providing some good fermentation result, but they're also helping the plant in the growth promotion. So this is where we have produced our bioorganic fertilizer. I think there's some issue. I'll have to, okay. Give me a minute. I have to reshare, sorry. I think I have to stop the sharing and reshare my slide. It's giving some issues. Okay, so this way we were talking about the bioorganic fertilizers. I have to skip it. Along with it, give me a minute, please. Can you please confirm me if you can see my screen now?
Yes, ma'am, we can see your
Okay, thank you.
screen.
Thank you. Okay, so we were talking about our biofertilizer, and okay, then I just want to conclude the work where we have what we have studied in our lab. We have worked upon the plant responses, the important responses in the plant, which are very important in mitigating or alleviation of the abiotic stresses. These are ROS scavenging, membrane stability, and osmo protection. Along with it, there are some things which are happening above the ground. These are the shoe targets. Most important are the stomatal regulations and the hydraulic conductance. And in the root, we have to control the ethylene production and oxygen level, and abscisic acid plays a very important role, the hormonal balance. And then along with it, we also have to maintain the root zone water availability. If we are able to control or target all these things, these help the plant in alleviation of the abiotic stresses, particularly the drought and the salinity. These are some of the awards and honors which have been received by me and my lab. This is a recent one where I have received the first Professor Dr. Abdul Hamid Gold Medal in the field of applied and environmental microbiology. And these are some other awards and honors which I have received from my university, like I have received some research award letters, the research incentives. Along with it, I'm also the president of Pakistan chapter of Asian PGPR, Society for Sustainable Agriculture. If anyone of you want to join it or any other information, I'll drop my email address. You can contact me at any time. We are now, this is also some other ways we are working upon the bioorganic fertilizer and the silicon nanobiofertilizer. These are also some of my other nominations. And by this, in the end, I'd like to thank particularly, most importantly, Professor Dr. Askari Banu, who had been my mentor, my PhD supervisor. And with what I am today is all because of her guidance. And I'm also thankful to all my lab fellows from my PhD lab, and also to all the colleagues which are working still together with me. And I'm also particularly thankful to the students who are working in my lab, my PhD and my MPhil students, and all the contributions of my lab. They're because of them. They're the one who are working day and night in my lab, and they're the one which are responsible for all these studies. So I thank you all for your time and listening to me. If there's any question, I'll be happy to answer.
Dr. Noshin, it was an honor having you here. Thank you so much for your informative talk. I will surely read the papers that you have mentioned, and it always is a pleasure to follow teachers' footsteps. Thank you very much.
Thank you very much.
Thank you.